Sessoms, and I'm a researcher at the University of Minnesota. I'm in the Department of Horticultural Science. I'm going to give you a quick update on the project titled Evaluation of the Cold Tolerance of Fescue Cultivars after de-hardening events. And I would like first to thank the Ontario Turfgrass Research Foundation for their support in our research. Before diving into our project, I really would like to thank my team. So I'm working with two different team members, Eric Wilkins, a professor at the University of Minnesota, Dominique Petrella, who was a researcher working at the University of Minnesota and moved on to assistant professor position at ATI, Ohio State University. Myself, a researcher at the University of Minnesota, and we have also hired an undergraduate student, Mary Knitting, who is helping tremendously on the different part of this project. Before diving into our experiments, we needed to request cultivar from the different seed companies. And to choose a cultivar or which company to contact, I went on the website, the National Turfgrass Evaluation Program, and I found which available cultivar were present and which seed company I needed to contact. I ended up with around 79 different cultivars from almost more than 10 different seed companies. So I need really to thank Mountain View Seeds, Alias Seeds, Pure Seed, DLF, Barenberg, Smith Seed Services, ProSeed, Grassland Oregon Seed, Pig Plant Genetics, Columbia, and Scott for their support in our research program. Before starting any de-hardening experiment, we needed to determine the freezing tolerance of all of cultivar. And for that, we did the freezing stress LT50 for all cultivar. So what is the LT50? Really, this is the median lethal temperature for each tested cultivar. And this result will indicate what is the freezing tolerance for each cultivar. So in this tray, for each line, we seeded one cultivar, and we seeded all cultivar, and the trays were moved for 10 days in the mist house until we had almost 90% of germination. Then we selected 3 random genotypes for each cultivar, and we moved like 1 genotype per cone. The cone were placed for 30 days in a greenhouse. The condition of the greenhouse were a 16-hour day length, with temperature ranging from 25 degrees Celsius during day and 20 degrees Celsius at night. We applied fertilizer 3 times with a concentration of 200 ppm of nitrogen. The plants were not clipped during that period. After, plants were moved for 14 days in a cold room with a 10-hour day length that had like a light intensity of 100 micromole, an average temperature of 2 degrees Celsius. During that time, plants were not fertilized, non-clipped, but they were watered as needed. Then we moved the plant in a 48 hours in a freezer. We tested 5 different freezing temperatures, -8, -12, -14, -16, and -20 degrees Celsius. The freezing program was up to -2 degrees Celsius for a soak, followed by the freezing stress with a ramp down of 1 degree per hour and 1 hour at temperature of interest, followed by a ramped up. After the freezing stress, plants were moved back to the cold room for 24 hours and then placed back in the greenhouse for a 14 days recovery. During that recovery, plants were fertilized, watered, and clipped as needed. After 15 days of recovery, we rated the plant. When the plant was dead inside a cone, we put a 0, whereas when the plant was alive, we gave it a 1. So here is the result of the first run. This is -8, where we had like almost a 100% survival, -12, -14, -16, and -20. And you can see that the lower is the temperature, the lower like the reduced freezing tolerance is happening. We repeated that experiment 3 times, and with all the rating, we were able to create this graph with the LT50 for all cultivar tested. So the average LT50 was minus 10.96 degrees Celsius. The maximum was minus 9.88 degrees Celsius. The minimum was minus 12.37 degrees Celsius. So here for EXP8PC and TD2, these minus like around close to minus 10 LT50 indicate that their freezing tolerance are low. And the lower is the LT50 indicates the higher is the freezing tolerance. So K18 WB1 and Raptor LS had the highest freezing tolerance. But what was interesting when we look at all the cultivar, this is that we observe variation in freezing tolerance that existed for all seed companies. And for example, DLF had like a LT50 of 10 minus 1031 to LT50 of minus 1161. So we decided based on this result that the LT50, the freezing temperature that we will use in our de-hardening experiment for the ghost chamber condition will be minus 12 degrees Celsius. So the third step was really to start our de-hardening experiment in ghost chamber condition. And this experiment is currently running. So first, what we had to do is really record the temperature at the University of Minnesota on campus in fall 2020 and fall 2021 to create our fall program. We ended up with like 4 different programs. We have Flow 1 that is run for 30 days with a day length of 13 hours, a light intensity of 300 micromole, an average temperature of 23 during the day and 12 at night. Then we move to Flow 2 for 7 days with a day length of 11 hours, a similar light intensity, average temperature of 20 and 11 degrees Celsius. We have after Flow 3 during 14 days with a day length of 10 hours, a light intensity of 300, and a day of 6 degrees Celsius and night of 0 degrees Celsius. Finally, we have Flow 4 with that is run for 7 days with a day length of night, a light intensity of 300, day of 0 degrees Celsius and night at minus 5. Then the plant will be moved to a program that we call Snow that will mimic a snow cover for 8 days. During that time, the day length will be of 8 hours, the light intensity of 50 micromole with average temperature of minus 5 during day and night. And this light intensity was really chosen because my colleague Dominique Petrola had sensor under the snow the previous winter and observed that the light intensity really could range between 20 and 70 micromole during the day. Finally, we'll have a de-acclimation program or de-hardening program that can last between 1 day or 5 days with day lengths of 12 hours, light intensity back to 300, day temperature of 12 degrees Celsius and night of 10. So how we decided, that all cultivars will be tested for the experiment, but because we have a lot of cultivar, we'll have 1 genotype per cultivar for each run, and we are planning to have this experimental setup run 4 times to be able to do statistical analysis. We have 3 treatments, a control that will never go under de-hardening. So basically the control plant, the control cones will go in the freezing stress after just before the end of the snow program. Then we'll have the 1 day of de-hardening where the plant will only go through 1 day of like this de-acclimation program. And finally, we'll have 5 days of de-hardening. And so why do we have 5 days of de-hardening? This is because we observe that the seasonal fluctuation where de-hardening could happen in field condition has been shown to last at least 5 days during March in northern United States and Canada. Then after this de-acclimation period, and for the control, so plant will be will go to a freeze stress in the freezer with a temperature of -12 degrees Celsius for 48 hours, then move back to the greenhouse, recovered, and rate with a 0 to 1 as for the LT50 experiment. So we have already started 2 runs for the experiment. We have week 1, week 2, week 3. We have like all of our cultivar in week 1, all of our cultivar in week 2, and all our cultivar in week 3. So this one will be the control, for example. This work will be the 1 day of deacclimation, and this one will be the 5 days of deacclimation. Then we have the second run with the plant, like each run is staggered to be like 15 days apart with rack 1, rack 2, rack 3, and the different treatment control, 1 day of declimation, 5 days of deacclimation. So we are also doing a field experiment where we'll try to do a de-hardening experiment. So for that, we are not going to test all the cultivars because of like space issue. So we are going to test up to 40 different cultivars. And this is a cultivar chosen and tested will be in function of their LT50, the best and the worst, but also the seed availability from the seed companies. We have 3 blocks and that have like the 3 different treatments, and each plot will be seeded at 2 pure life seed per square centimeter. We have also a couple of sensors for that experiment. So here, this is lab jar that would be placed in the soil, really to record the soil temperature for the whole experiment. And I try to place all these like lab jack at a 10 centimeter depth. Then we have also the ability to record O2 and CO2 for all rep, but only for block 1. And finally, for the de-hardening period, we will have like hobo sensor that will be placed really below the surface to record the temperature, the soil temperature during the de-hardening experiment. They have the ability to communicate with a cell phone via Bluetooth, so we can measure really if the deacclimation is happening or not. And the field experiment was already started this September, seeded like September 8, 2021, and we will plan to repeat that experiment next fall. So here, this is our experimental setup or field, north on the left side of the picture, south on the right side of the picture. We have block 1, block 2, and block 3 with a gas sensor on the north side of all reps for block 1, and the lab jack present in the middle of the rep for block 1, block 2, and block 3. And we'll also have the hobo sensor placed in each of like these this rep when we'll deacclimate them. For the deacclimation period, for the 1 day or 5 day, because we also have a control that will not be deacclimated, will in fact, our methodology will be to place like a cover on top of the rep and have like these heater that have like a thermostat and that can really desoil the soil. The hobo really will help us to see if like the thawing, is happening. So basically, the 1 day or 5 days will maybe be longer because we are thinking that we need 48 hours to have like the soil thawed. The experiment is really planned by the end of February, and we'll rate the survival and spring green up when in spring. So finally, I wanted to mention that we also had like such a nice preliminary result from the LT50. We have like created this time course experiment that we a nice addition of the observation from the LT50 experiment. So what happens, you see that between -12 and -14, we really observe a variation of survival between the different runs. So basically what happens is even if greenhouse conditions are kind of controlled, we had a very hot summer and ending up by stunning the growth of like the plant in for run 2 and run 3. and really observe a difference of survival between run 1, run 2, and run 3. So it led to the assumption that maybe the development stage of the plant, height, number of tiller, number of leaves, can influence the freeze-stress survival. So we did a very small, very quick experiment, a time course experiment, and where we choose like 2 of the worst and 2 of the best cultivars based on the LT50. And we are doing a time course experiment. So plant will be submitted to a freeze stress 24, 38, 52, and 60 days after seeding. The freezing temperature tested will be minus 14 degrees Celsius. And also what I want to mention, that's between the greenhouse and the freezing, we'll have like a 2 weeks cold acclimation. And also before moving the plant to the cold room, we'll really measure the height, the number of tiller, and number of leaf, and see if the development stage can have an influence on the tolerance to freeze. And that will be very important because it can really help us to really manage better our experimental setup for freezing tolerance. So really, that's it for all of the experiment we are currently running. What I didn't mention is like how we are going to disseminate or disseminate our data. Basically, we are planning really to showcase our data through the blogs. We would like also to present like our field experiment during field days. We would love to go to the grower field days and present our data either in Northern United States, in Canada. We are also going to present all the results, all of our first year and second year results at the ASA, SSSA, and CSSA meeting in 2022. And we are already starting to write a publication for the LT50 experiment, and we'll be aiming for a second publication for the de-hardening experiment. So there will be a lot to share, a lot of exciting and new results. So I gave you a lot of information. It was very fast. It was a lot of observation, results, etc. If you have any question, any follow-up question regarding our experimental setup or ideas, what we are doing, please do not hesitate to contact me at my email address, fsessom@umn.edu. Again, I thank you for your attention.